Welcome to G Fun Facts Online. Today, we are deconstructing a fundamental misconception about human biology. For decades, the human brain was viewed as a static organ, a biological computer whose hardware was finalized in childhood, destined only to decline with age. We now know this is false. The brain is a dynamic, shifting landscape, capable of profound physical reorganization throughout the entire lifespan. This quality, known as neuroplasticity, is not merely a response to learning or trauma. It is a relentless, daily adaptation driven by a powerful internal chemical orchestra, the endocrine system. This is the story of neuroendocrine adaptation, the scientific frontier where hormones reveal themselves not just as regulators of metabolism or reproduction, but as the master architects of neural circuitry. From the reshaping of the maternal mind to the synaptic pruning caused by chronic stress, hormones do not just signal the brain, they rewire it. They alter dendritic spine density, modulate the birth of new neurons, and resize entire brain regions to suit the environmental demands placed upon us. This is the story of how the chemical self constructs the electrical self. To understand how a molecule floating in the bloodstream can restructure a thought, we must look at the cellular machinery. Hormones like cortisol, estradiol, and insulin do not simply knock on the door of a neuron. They often possess keys to the very genetic control room of the cell. The influence of hormones on the brain occurs through two primary pathways: genomic and non-genomic actions. In the genomic pathway, steroid hormones such as estrogen and cortisol behave like ghosts passing through walls. Because they are lipophilic, or fat-loving, they slip effortlessly through the neuron's fatty outer membrane. Once inside, they bind to receptors in the cytoplasm and travel into the nucleus. There, they act as transcription factors, literally reading sections of DNA to increase or decrease the production of specific proteins. These proteins might be the building blocks of new synaptic terminals, or the receptors that make a neuron more sensitive to electrical signals. This is a slow, deliberate process of rebuilding the neuron from the inside out. The non-genomic pathway, however, is rapid, occurring in seconds or minutes. Here, hormones bind to receptors on the cell surface, triggering rapid signaling cascades known as kinase pathways. These can instantaneously alter the excitability of a neuron, making it more or less likely to fire. At the structural level, the most visible impact of these mechanisms is on dendritic spines. These tiny mushroom-shaped protrusions on the branches of neurons are the receiving docks for synaptic signals. A single neuron may have thousands of spines, each one a potential connection point. Hormones regulate the density and shape of these spines. Under the influence of estrogen, for instance, the hippocampus, the seat of memory, blooms with new dendritic spines, creating a dense forest of connectivity. Conversely, under the siege of chronic stress hormones, these spines wither and retract, thinning the forest and severing the lines of communication. This brings us to the hypothalamic pituitary adrenal axis, or the HPA axis, the body's primary alarm system. Its end product, cortisol, is perhaps the most potent rewiring agent in the modern human experience. In the short term, cortisol is a performance enhancer for the brain. During an acute crisis, such as a near-miss car accident or a high-stakes presentation, cortisol floods the system. It sharpens attention and mobilizes glucose for immediate neural energy. It acts specifically on the amygdala, the brain's threat detection center, to etch the memory of the danger deeply into the neural fabric. This is an adaptive survival mechanism. The brain prioritizes remembering what could kill you so you can avoid it next time. This flashbulb memory effect is why you can remember exactly where you were during a traumatic event, but might forget what you had for lunch yesterday. However, this system was designed for sprints, not marathons. When stress becomes chronic, due to relentless work pressure, financial insecurity, or systemic trauma, neuroendocrine adaptation turns maladaptive. The constant bath of cortisol begins to poison the very structures it once protected. The most significant damage occurs in the hippocampus. This region contains a high density of glucocorticoid receptors which bind to cortisol. Chronic overstimulation of these receptors triggers a process of excitotoxicity and dendritic retraction. The neurons in the CA3 region of the hippocampus begin to shed their dendritic spines and eventually die off. MRI studies have consistently shown that individuals with Cushing's disease, who have excess cortisol, or those with severe untreated PTSD, possess significantly smaller hippocampal volumes. The result is a fragmentation of narrative memory and a loss of context. The brain loses its ability to distinguish between a past threat and a present safety, trapping the individual in a loop of reactive anxiety. While the hippocampus withers, the amygdala does the opposite. Chronic stress causes the amygdala to grow larger and more reactive. Dendritic trees in the basolateral amygdala expand, creating more connections. The result is a brain that is physically wired for fear, a hypervigilant state where neutral stimuli like a loud noise or a stern look are misinterpreted as severe threats. Simultaneously, we see a disconnection in the prefrontal cortex, or PFC. The PFC is the CEO of the brain, responsible for logic, impulse control, and emotional regulation. It normally keeps the primitive amygdala in check. Chronic stress severs the synaptic connections between the PFC and the emotional centers. The cables are effectively cut. The CEO loses control of the security guard. This neuroendocrine remodeling explains the brain fog, impulsivity, and emotional volatility often seen in burnout and chronic anxiety disorders. Moving beyond stress, we find that sex hormones are equally powerful sculptors. For decades, science relegated sex hormones like estrogen, progesterone, and testosterone to the realm of reproduction, assuming their influence stopped at the neck. We now know that the brain is a sexual organ, at least in terms of its receptor density. These hormones act as potent, neurotrophic factors, protecting neurons from death and stimulating the growth of new connections. The hypothalamic pituitary gonadal axis, or HPG axis, drives massive remodeling events at key life stages: puberty, pregnancy, and menopause. During puberty, a tidal wave of gonadal hormones triggers a massive reorganization of the brain. Interestingly, the brain actually loses gray matter volume during this time, a process known as synaptic pruning. This is not a loss of function, but a gain in efficiency. The hormones signal the brain to cut away weak, unused connections to strengthen the major highways of communication. It is a refinement process, transitioning the brain from a child's sponge-like absorption state to an adult's focused executive state. In adult females, the brain is not static from month to month. Research on the hippocampus has shown that dendritic spine density fluctuates in rhythm with the menstrual cycle. When estrogen levels peak around ovulation, spine density in the CA1 region of the hippocampus increases significantly, enhancing verbal memory and spatial reasoning. As progesterone rises and estrogen falls, these spines are pruned back. This cyclical breathing of the brain suggests a flexibility in cognitive strategies across the month, optimizing the brain for different types of tasks at different hormonal phases. Perhaps the most dramatic example of neuroendocrine adaptation is pregnancy, often called matrescence. The flood of hormones during gestation is unprecedented. Estradiol levels rise hundreds of times above normal. This orchestrates a radical restructuring of the maternal brain. MRI studies reveal that pregnancy leads to a substantial representation in gray matter volume in regions associated with social cognition, such as the theory of mind network. Far from being a deficit, the so-called mommy brain myth, this volume loss is a focused specialization. The brain is streamlining its circuits to become hyper-efficient at reading the infant's cues, predicting needs, and detecting threats. This restructuring predicts the quality of maternal attachment. The more the brain changes, the deeper the bond. These changes can last for at least 2 years postpartum, and some evidence suggests they may be permanent, leaving a fingerprint of motherhood on the brain's architecture. Later in life, as women enter menopause, the precipitous drop in estrogen represents a bioenergetic crisis for the brain. Estrogen is a master regulator of the brain's glucose metabolism. When it fades, neurons in the hippocampus and cortex can struggle to generate energy, leading to temporary cognitive instability, manifesting as hot flashes, brain fog, and memory lapses. This is a critical window of vulnerability. However, the brain is resilient. It eventually undergoes a compensatory adaptation, shifting its metabolic machinery to utilize ketone bodies and other fuel sources, settling into a new equilibrium. We must also consider the metabolic hormones. The brain consumes 20% of the body's energy despite being only 2% of its weight. It is ravenous, and therefore it listens intently to the hormones that regulate energy balance: insulin, leptin, and ghrelin. Insulin receptors are densely packed in the hippocampus. Insulin does not just regulate blood sugar, it regulates synaptic plasticity. It supports the survival of neurons and the formation of memories. A growing body of research suggests that Alzheimer's disease may be, in part, a form of insulin resistance in the brain, often termed type 3 diabetes. When neurons become deaf to insulin's signal due to chronic metabolic dysfunction, such as that caused by a high-sugar diet, they can no longer repair their synapses, leading to cognitive decline. Similarly, leptin, the satiety hormone, and ghrelin, the hunger hormone, have receptors throughout the brain's reward centers. They do more than tell you when to put down the fork. They alter the value of external stimuli. High levels of ghrelin, which indicate hunger, actually rewire the hippocampus to be better at spatial learning. This is an evolutionary adaptation to help a starving hunter remember where the food is. Conversely, leptin promotes long-term potentiation in the hippocampus. Obesity, which often involves leptin resistance, can impair this plasticity, creating a disturbing link between metabolic health and cognitive flexibility. The chemistry of connection is driven by social hormones, oxytocin and vasopressin. These are unique because they act as both hormones in the blood and neurotransmitters in the brain. They are the architects of social circuitry. Oxytocin, released during touch, breastfeeding, and orgasm, quiets the amygdala. It inhibits the fear response and strengthens the connection between the emotional centers and the prefrontal cortex, promoting trust and social synchronization. In animal models, oxytocin can change the auditory cortex of a mother mouse to be specifically tuned to the frequency of her pup's distress call. It literally rewires the sensory processing centers to prioritize the social bond above all other noise. Vasopressin, closely related to oxytocin, is vital for social memory and territoriality. In famous studies of voles, the difference between a monogamous, pair-bonding prairie vole and a promiscuous meadow vole comes down to the distribution of vasopressin receptors in the brain's reward system. By genetically altering these receptors, scientists can rewrite the social behavior of the animal. This implies that our capacity for loyalty, monogamy, and tribalism is deeply rooted in the neuroendocrine tuning of our reward circuits. The story of neuroendocrine adaptation gets even deeper when we consider epigenetics, the ghost in the machine. Hormones can permanently tag DNA, switching genes on or off for the long term. This is the mechanism behind transgenerational trauma. If a parent experiences severe chronic stress, their HPA axis dysregulation can lead to epigenetic changes in their sperm or egg cells, or the uterine environment. High levels of glucocorticoids can methylate genes related to stress response in the developing fetus. The result is an offspring with a brain already wired for hypervigilance and high anxiety before they have taken their first breath. The hormone rewires the brain of the next generation, preparing them for a dangerous world that the parent experienced. The implications of this science are profound. If the brain is plastic and responsive to hormones, it means we have agency. We can influence the hormonal bath our brains soak in. Physical activity acts as endocrine therapy. Muscle contraction releases myokines, which cross the blood-brain barrier and stimulate the release of BDNF, or brain-derived neurotrophic factor. BDNF is essentially miracle-grow for the brain, triggering neurogenesis in the hippocampus and protecting against cortisol-induced atrophy. Sleep is another pillar, as it is when the neuroendocrine system resets. Growth hormone acts during deep sleep to repair tissue, while the glymphatic system flushes out metabolic waste. Protecting sleep is protecting the hormonal regulation of brain structure. Finally, positive social interaction releases oxytocin, which buffers the corrosive effects of cortisol. We can literally protect our brain structure through the maintenance of healthy relationships. In conclusion, we are not fixed entities. We are a fluid collaboration between our genetic blueprint and our chemical environment. Neuroendocrine adaptation is the mechanism of this fluidity. It is the bridge between our lived experience and our biological structure. From the pruning fires of puberty to the protective rewiring of motherhood, and from the ravages of chronic stress to the healing power of social bonding, hormones are the silent sculptors of the self. Understanding this interplay offers us a new map for mental health, one that treats the brain not just as a machine of electrical impulses, but as a garden dependent on the chemical soil in which it grows. By tending to our endocrine health through diet, stress management, connection, and movement, we are effectively acting as the architects of our own brains. Thank you for listening to G Fun Facts Online.